Beneath every breath of air and droplet of water lies a war zone invisible to the human eye. It's not a metaphorical war, nor one of ideology or borders, but a relentless biological clash that has shaped life on Earth for eons. The foot soldiers in this ancient struggle are not humans, nor animals, but entities stranger than fiction, viruses that prey exclusively on bacteria. They're called bacteriophages. Their name means bacteria eaters, though they do not consume in the traditional sense. They dismantle, disassemble, repurpose. They are not alive in the way we understand life. No metabolism, no nervous system, no awareness. But they are not inert either. Each one is a microscopic machine built solely for the purpose of identifying, invading, and destroying bacterial cells. They are absurdly abundant. If you could somehow gather all the bacteriophages on Earth and stack them, their combined length would stretch beyond the Milky Way. Trillions swarm in the oceans. Billions drift across your skin. They outnumber every other organism on the planet by a margin so wide it's almost pointless to quantify. Their design is unsettlingly geometric. A crystalline head containing strands of DNA or RNA, a shaft that resembles a hollow drill, and spindly leg-like fibers that clutch onto bacterial hosts with eerie precision. Once a phage locates its target, it locks on and injects its genetic payload, hijacking the cell's machinery and turning it into a viral factory. The result? A singular bacterial cell becomes a nursery for dozens, sometimes hundreds, of new phages. When replication is complete, a final enzyme ruptures the host from within, releasing a new wave of viral killers. This is not an isolated event. It happens constantly, at an astronomical scale. In the world's oceans alone, up to 40% of all bacteria are destroyed by phages every single day. But despite their ruthless efficiency, phages pose no threat to us, not directly. They are too specialized. Each phage is tuned to recognize only one species or strain of bacteria. The war they wage is targeted, precise, and ancient. We've lived alongside this hidden war forever, mostly unaware. And now, for the first time in human history, we may be ready to harness this microscopic violence. What if the very organisms that commit bacterial genocide could be repurposed, not to harm us, but to protect us? In the early 20th century, a new era began with a drop of mold juice on a forgotten petri dish. It wasn't elegant science, but it changed medicine forever. The discovery of penicillin transformed humanity's relationship with bacteria. For the first time, microscopic infections, once deadly in their silence, could be stopped. And so began our golden age of antibiotics. Penicillin was only the beginning. In the decades that followed, we extracted and synthesized a multitude of antibacterial agents, each one more refined and targeted. Infections that once killed millions, strep throat, tuberculosis, pneumonia, became manageable or even trivial. The balance of power between humans and microbes had shifted dramatically. But power breeds complacency. We began deploying antibiotics with increasing frequency and decreasing caution. A minor fever, a precautionary treatment, an additive in livestock feed. We wielded this chemical sword against problems both real and imagined. And the enemy, being alive, did what living things do. It adapted. Bacteria are deceptively simple, but evolution favors simplicity. Their rapid reproduction and high mutation rates meant that antibiotic resistance emerged far sooner than we anticipated. In just a few generations, mere days or weeks for bacteria, random mutations allowed some strains to survive exposure. These survivors multiplied, passed on their defenses, and shared them via gene-swapping mechanisms that blur the lines between species. By the 21st century, we had created monsters. Superbugs, as they're now known, are bacterial strains immune to nearly every antibiotic in our arsenal. They colonize hospitals, cling to ventilators, thrive in wounds, and outlast even our most aggressive therapies. For patients infected with these resistant strains, treatment becomes a desperate guessing game, often ending in death. The numbers are no longer small. In the United States alone, over 23,000 people die each year from infections caused by resistant bacteria. Globally, projections estimate that by 2050, superbugs could kill more than 10 million people annually, surpassing cancer. These aren't speculative doomsday forecasts. They're already unfolding in real time. Patients have died from urinary tract infections that no drug could cure. Children have succumbed to routine cuts. Entire hospital wards have been locked down because of a single resistant strain. It's a haunting reversal. For decades, we had the upper hand. Now, we're watching the clock run out on a medical revolution we assumed would last forever. But just as the natural world once gave us penicillin, it may yet offer a second chance. This time, not from fungi, but from viruses. There's a strange symmetry in the idea. The same microscopic killers that slaughter bacteria by the trillions may become our new allies. Not because they care, not because they're intelligent, but because they've been locked in a ceaseless evolutionary war with bacteria long before we ever arrived. If antibiotics were our broad-spectrum bombs, then phages might be our surgical tools. But how does one control a predator that exists to destroy? And what makes these tiny killers different from the weapons we've already exhausted? Imagine a battlefield where every weapon knows its target's name, genetic signature, and biological vulnerabilities. This is not science fiction. It's microbial reality. While antibiotics act like indiscriminate firestorms, scorching entire bacterial ecosystems, including those essential to human health, bacteriophages operate more like snipers. They don't just kill bacteria, they kill the right bacteria. This precision is the result of billions of years of coevolution. A single phage typically hunts one bacterial species, sometimes even a specific strain. It recognizes its prey by the unique molecular shapes on a bacterium's surface. When contact is made, the phage deploys a complex mechanism resembling a spring-loaded syringe. The tail contracts, puncturing the bacterial membrane, and viral genetic material is injected directly into the host. What follows is ruthless. The bacterial cell is reprogrammed into a viral foundry, churning out phage components until it can't hold them any longer. Then, with the help of an enzyme engineered by the virus, the bacterium's wall is ruptured from within. The cell bursts, releasing a swarm of new phages into the surrounding environment, each ready to repeat the process. Now, imagine using this process not in seawater or soil, but inside a human body, intentionally. It sounds counterintuitive. Introducing viruses into a sick person goes against everything modern medicine has taught us. But here's the crucial distinction. Bacteriophages are harmless to humans. They don't recognize our cells, don't interact with our biology, and are ignored by our immune systems. Every day we coexist with trillions of them, on our skin, in our mouths, in our gut, without consequence. In fact, that's what makes them so promising. Unlike antibiotics, phages leave beneficial bacteria untouched. They eliminate only the target pathogen and then vanish. And if the bacteria adapt, the phages adapt too. Their evolutionary dance with bacteria is never static. When one develops resistance, the other counters. It's a dynamic, built-in upgrade system that pharmaceuticals can't match. But the advantages go even deeper. In some cases, when bacteria evolve a resistance to phages, they inadvertently sacrifice their resistance to antibiotics. It's a molecular trade-off, a trap. Scientists are now exploring combined therapies, applying phages alongside traditional antibiotics to force bacteria into a biochemical catch-22. If the bacteria defend against one, they become vulnerable to the other. In one extraordinary case, a man suffering from a chronic antibiotic-resistant infection had phages directly administered into his chest cavity. The bacteria, Pseudomonas aeruginosa, known for its defiance, had survived for years, evading every treatment. But when the phages were introduced, paired with antibiotics, the infection began to collapse. Within weeks, the bacteria were gone. This is more than anecdote. The therapeutic potential of phages is beginning to move from last resort desperation to frontline innovation. Clinical trials are underway. Laboratories are mapping phage bacteria matchups with the precision of genetic surgeons. The field is still experimental, but it's gaining ground, and fast. Still, phage therapy isn't plug and play. Each infection may require a custom phage cocktail. Manufacturing, testing, and approval systems for such individualized treatments remain complex and expensive. For now, most major pharmaceutical companies remain hesitant, unwilling to invest in a field without standardization or guaranteed return. But the stakes are too high to ignore. Could these ancient, indifferent killers become the scalpel we need in a world where our blunter tools have failed us? It may sound like science bending into paradox, using viruses to treat disease, but the future of infectious medicine might depend on precisely that contradiction. In a world growing increasingly resistant to conventional drugs, phages are not just promising. They may be essential, but progress demands more than good ideas. It requires systems that can support them. At present, phage therapy exists in a patchwork landscape of compassionate use cases, experimental treatments, and isolated success stories. Despite encouraging results, the field faces considerable inertia. Major pharmaceutical companies are slow to adapt, wary of the economic uncertainties tied to personalized, biologically active medicine that lacks one-size-fits-all scalability. Why? Unlike broad-spectrum antibiotics, which can be manufactured, bottled, and sold in bulk, phages are specific. What works for one infection may be useless against another. Each patient, in theory, might require a different viral solution. This makes mass production, distribution, and profit forecasting extremely difficult. It's not just a scientific challenge, it's a logistical and economic one. And yet, momentum is building. In 2016, the largest clinical trial involving phage therapy began in Europe, aimed at standardizing treatment protocols and establishing safety benchmarks. More countries are opening regulatory pathways for emergency use. Research centers are developing vast phage libraries, matching viral strains with known bacterial threats to streamline treatment. But even more compelling is what phages represent beyond the lab, a new paradigm of medical thinking. For nearly a century, we relied on brute force, antibiotics that obliterate anything microbial in their path. But that era is waning. The microbial world is vast, ancient, and deeply interwoven with our biology. Our gut alone contains more bacterial cells than human ones, forming a delicate ecosystem crucial to our health. Carpet bombing it every time we sneeze has consequences: metabolic disorders, autoimmune issues, resistance spread. Phages offer a chance to move from destruction to precision, not to eradicate microbial life, but to shape it, to guide it, to protect what benefits us while targeting what harms us. That's a leap not just in medicine, but in mindset. And it opens the door to further possibilities. Could phages be engineered to deliver genetic therapies? Could they be used to manipulate bacterial populations in agriculture? Reducing our reliance on antibiotics in livestock, might they help treat conditions indirectly related to bacterial imbalances, like inflammatory bowel disease, or even certain cancers? These questions are no longer theoretical. Teams around the world are already exploring them. And though many hurdles remain, legal, technical, cultural, the path forward is being paved with growing urgency. The irony is difficult to ignore. We once viewed viruses as our purest enemies. They cause pandemics, ruin lives, evade our best efforts to contain them. But now, in the folds of that fear, we're discovering unlikely allies. machines of destruction, fine-tuned by evolution, ready to serve a different purpose. We will need to rethink our categories. Not all viruses are our enemies. Not all bacteria are our foes. And not all solutions arrive looking like saviors. Sometimes the thing that saves us is the thing we feared most, reimagined, repurposed, and understood with new eyes. As the post-antibiotic age looms on the horizon, the quiet war between bacteria and phages continues, as it has for billions of years. This time, though, we might choose a side. And with that choice, we may find ourselves saved by the most efficient killers nature ever made.